I'm an assistant professor, and my lab focuses on the application of mass spectrometry and experimental genetics to dissect immune regulation, especially in the context of cancer and infectious diseases. A lot of our work involves mass spectrometry analyses of immune cells, and often these are either harvested from humans or in animal models. And because these are very precious samples, they're very expensive to sort in large quantities, it's extremely important to have high sensitivity analyses of these samples. So we spend a lot of our time optimizing that. And as part of that pipeline, we've begun using the EvoSep system. A couple of projects I'd like to highlight include our exhaustion project in T-cell exhaustion, which is a collaboration with our partners, the Hakim Lab here at Emory, and also our own project studying pathway mapping using experimental genetics paired with proteomic analyses. Absolutely. So a lot of our recent work has focused on T-cell regulation, and we've recently completed 2 projects in the realm of T-cell regulation. The first involves studying T-cell exhaustion in the LCMV mouse model, which is an extremely powerful and well-characterized model for understanding T-cell exhaustion. The beauty of this model is that you can have either an acute or a chronic infection in the mice, and thus purify from those mice antigen-specific T cells, either following the effector memory trajectory or the exhaustion trajectory. And by comparing these 2 sets of T cells, we can identify the proteomic landscape of T cell exhaustion. To our knowledge, we're the first lab to perform this phosphoproteomic analysis using the LCMV model system. The other type of analysis I'd like to highlight for you is our pipeline doing pathway analysis using phosphoproteomic and protein abundance analyses coupled with experimental genetics in primary T cells. And the beauty of this is we can actually perform saturating mutations with CRISPR-Cas9, selectively remove components of those pathways or mutate residues in the proteins mediating these pathways. We can add interferon, and then we can see what changes downstream of that stimulus as a consequence of our perturbations. And so by marrying experimental genetics to proteomic analyses, we can start to take apart signaling pathways relevant to immune function and other functions in the body. So in using the EvoSep, what we've noticed is 3 main things: robustness, sensitivity, and throughput. So in terms of robustness, we don't have to worry about this instrument going down. We generally don't have to worry about fluidic problems. We don't have to worry about anything. It just works. We run hundreds, thousands of samples with this instrument for months, and it just keeps going. So that's been wonderful. In terms of throughput, the ability to run on our rig, we can run up to 100 samples per day. On newer EvoSeps, I understand it's even greater numbers. Our observation with our setup is that throughput is reproducible. We see reproducible sample analyses, and so we've really enjoyed that experience. In terms of sensitivity, the ability to concentrate samples on EvoSep tips and avoid any loss associated with putting them into microwell plates has really helped us to boost sensitivity in low input contexts.
Coming from a proteomics background and usually using very classical LC systems, I'm used that LC systems can be quite time consuming and problematic. Having the opportunity to work with an EvoSep really made life significantly easier. It's no much longer to worry about if a sample really injects or if we have like column variation or like injection variation. We can just spend more time doing our research and forgetting about the LC part of it. So overall, this really helped us to work with smaller sample amounts, because nowadays we can actually really make sure that every sample counts, which we actually process. And for example, also in our LCMV sample, in our LCMV study, we could work with much less sample material than we would have potentially used or needed for our classical sample approaches.
So for example, our work on phosphoproteomics, Christian in the lab, he's deployed a new proteomic pipeline that we call SimplePhos. And the idea of SimplePhos is we can do phosphoproteomic enrichment in 96 well plates without any need for specialized liquid handling robots. Then we can take these samples, these phosphoproteomic enriched samples, put them directly onto EvoTeps, and inject them that way. So that way we concentrate those phosphoproteomic samples right on that tip and inject all of it. We don't end up with loss associated with adsorption to plastic. We don't have loss associated with dead volume in those microwell plates. So that's been fantastic for our workflow.
Prior to having the EvoSep system, we had to manually dry these samples and then resuspend them, fill them into vials, and then inject them into the mass spec. This not only required a lot of time, but also induced a lot of variation, just because of the additional handling steps, and obviously sample losses, because we had to transfer the samples between multiple vessels. So having the option to have an EvoSep and just directly loading the samples onto the EvoSep itself, or the EvoTips in this case, really made a drastic improvement in our time spent in the lab. For a lot of people who do routine analysis of certain types of sample, I definitely think an EvoSep makes it very easy to just do these samples. If people are doing a lot of method development and need a lot of tinkering or want to have the flexibility of their own gradient, obviously an EvoSep is maybe not the right choice. But I think as a just reliable workhorse, the EvoSep system definitely does the job.
Not only would I have. I find that EvoSep has this combination of robustness, throughput, and improvements in sensitivity that have really benefited our pipeline studying immune cells. And so for that reason, I do tell my colleagues in the mass spectrometry community that we've had really great experiences using this system. It's also ease of use, right? Because it has these predefined liquid chromatography parameters. You don't have to spend this time tweaking those different settings. And I think that's another thing I'd like to mention is moving forwards in mass spectrometry, I think it's very important to have this technology available to a wider community. I think it's still in the realm of specialists who understand mass spectrometry, the nuts and bolts of how these machines are built, the nuts and bolts of how to configure the analysis pipelines, how to process the data. On the liquid chromatography front, I think EvoSep has done an outstanding job of making liquid chromatography more straightforward and more accessible to users who may not be specialists in liquid chromatography. And then moving forwards into the mass spectrometry analysis software, I think there's been major advancements there in making the software much more user-friendly and approachable so that people who are not specialists in proteomics can process this data themselves and gain mechanistic insights into the biology, which is really what we're driving towards. We want to understand how biological systems are built. So the easier it is, and the more efficient it is, and the more robust it is, and the more reproducible it is to run proteomic workflows all the way from sample to LC to mass spec to analysis to insights. We want that to be as smooth as possible, and I think EvoSep is a great addition to that ecosystem.